So we were here, like we can use different uh, the way this personalized genomics can be used in different setups like long-term medical care, financial interest, family tree. So this one of the key use of this personalized genomics is using the family tree. Like you can find out your ancestors, like how they are. So most of the
This is
well,
a
this is not a
demonstration
problem
of
in India, but
Wirecast.
in US it's the biggest problem. You never know who's your grandparents are, who's your parents are, who's your mother, who's your father, or blah blah blah, everything. So people are like curious to know like what is their ancestry, like how that is related. Now, having said that, these are some of the important key players in the personalized genomics companies. So these are the companies, these are the commercial companies. So they do the
This
testing.
is a
Some of
demonstration
them
of
do the genetic
Wirecast.
testing. Some of them has recently been accorded a FDA approval to do the drug to drug the disease related association um, indications for the individual. So they can do blood test, sputum test, urine test, or stool test, or depending on, most of the time it is sputum or blood that is mostly used. So 23andMe is the biggest and the
This is a
foremost player
demonstration of
in personalized
Wirecast.
genomics. So just a little bit of background, if you don't know, is so this is a company that is created by Annie. So she's wife of Google's Sergey Brin. So now they are divorced, but she was a very good scientist in the initial stages, and her interest from the personal genomics stems from the last 20 years when whole this genome was not even sequenced. So at that time, so
This
she
is a
established a
demonstration
small
of
company
Wirecast.
with Sergey, and there is another partner to sell the personal genome. So the key, so their initial bet was to give the ancestry. So that is $99. If you go to, I will show you the. So this is basically this is the company's web page. So basically they give your whole genome for $99. So
This is a
they will send you a
demonstration
kit,
of
they will
Wirecast.
send you a vial, you put the blood or the sputum, depending on what you're trying to do, is send it back, and within like a week, they have the people, they have the set, everything is set. So they will profile your genome, all 23 chromosomes, and then send that, okay, this is what is happening is, but they are very careful, they don't predict. For example, if they have found out BRCA1 mutation, they will highlight it, but they will never say that, okay, you are liable to be susceptible to breast
This
cancer
is a
because
demonstration
of
of
the,
Wirecast.
liability issues. So they don't do any interpretation. So basically, they leave it to the user to interpret it, or go to a clinician, or do it themselves. So this is one of the key area where where they are leaders. Now their most selling point was ancestry. At that point, till like last 10 years, they have been doing a very good job in selling the ancestry tree. So they will make ancestry tree, phylogenetic
This
tree,
is a
and tell that,
demonstration
okay,
of
this may
Wirecast.
be these is the profile of your parents, grandparents, and blah 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 other things, and probably this is the geographical location, and this is your. In US, like it always happens, is say one parent is coming from Afro-Asian, another is Caucasian, Asian, Korean, Chinese, and all this, everything is mixed up. So there's no separate guidelines. So you can just kind of have a view like what is your ancestors are. That was very popular till today.
This is a
But what has
demonstration of
happened is FDA
Wirecast.
very recently they have approved for 6 tests. So they were doing this kind of testing for like 7-8 years. So what they were doing is they were sending that sample kit to the individual. Individual will send back a sample. So basically no clinician is involved. No one else is involved. It is directly to the consumer. So which we call it in short as a DTC. So 23andMe and DTC. So then FDA, someone complained to FDA. So FDA said, no, you cannot do that. So you
This is a
don't have the
demonstration
right
of
to
Wirecast.
directly sell to the customer and make the financial gains and tell that this is what you can do is say, you need the FDA approval. So they stopped it for 3-4 years, and then they have pretty nice team of attorneys. Again, at the end of the day, it is about money. So she has a lot of money. So they fought with the FDA and filed the FDA approval. So finally, then FDA approved 6 tests, which they
This
can
is a
send it directly to
demonstration
the
of
consumers
Wirecast.
without any clinician. So this is the biggest achievement in the history of the personalized genomics. But all 6 diseases, you can go online and figure it out, but most of them are neurological disorders, like Alzheimer, Parkinson's disease, and others. And so that's why the Parkinson's disease, one of the gene mutation is in Sergey Bin's, and his father has already Parkinson's disease full-fledged. So that's why he's
This is a
donating a lot of money, I think I
demonstration of
mentioned last time,
Wirecast.
to the Parkinson's disease foundations. That's one of the key way of funding in United States at least. So, so I think it is worthwhile if you just go and search 23andMe. So 23andMe name comes from 23 chromosomes, and basically the reality is you have 23 chromosomes and how it looks like in me. So that's basically the concept is. So now I think they both are separated.
This is a
So Annie is
demonstration
still
of
leading that one.
Wirecast.
So I will not go into further details. So another company is Navigenics. So this is one of the co-founder is David Agas. So he's from USC, and he's our collaborator. So we have this year we have also published with him, but he is he's a very good orator, and he can generate the money right sitting in this room in 10 minutes. So he has been given I think approximately
This is a
200 million by Larry
demonstration of
Ellison.
Wirecast.
Larry Ellison, everyone know? Larry Ellison, you should be knowing. Larry Ellison, he's a guy who has cloud computing. So he brought the concept of all the cloud computing and the informatics in all the world. So he's very, very famous, Larry Ellison. So he has I think 200 or 300 million dollars for this guy to establish his separate personalized genomic edition. Then Microsoft gave him the money. So he has
This
a lot
is a
of money.
demonstration
He's
of
making
Wirecast.
a separate institution and other things. But he is doing a pretty good job also. So he treats he works at Cedars-Sinai Hospital, which is nearby. He works at USC, in the USC hospital, but he also works at Cedars-Sinai. So Cedars-Sinai Hospital is very famous for treatment of celebrities. Most of the Hollywood actors, most of the rich people, they come there. Okay, so he mostly is associated with these big shots, and that's
This
why
is
he
a
gets
demonstration of Wirecast.
donations. Okay. Now, so this is the guy. So he established this company, but I don't think it is doing pretty good, but it is still existing because his priorities get changed. Like I think he's establishing his own Larry Ellison Translational Genomics Institute. He's establishing, but he is in too many things. So, but 23andMe, there's no match. All these companies on one side and 23andMe
This
on
is
one
a
side.
demonstration
Because
of
Annie has her
Wirecast.
focus, 100% focus on that company. Then there is color genomics. So this is a combined effort by Google, Twitter, Stanford, and MIT. So they basically combine most of the scientists from Stanford and MIT, and then Google, CEO, Twitter, they all contributing into this money. So the idea here is not to kind of scare you away like there's so much money is involved. So all
This
these
is a demonstration
these
of
companies, social
Wirecast.
media, and all these big shots, so they care about this kind of personalized genomics. That's the key point which I'm trying to make is, so it's not just a jargon of the scientists like me or anyone help, because we have a job to do and we keep on doing. So there is a lot of efforts on part of the social media as well as this. So now if you see, now we are living in a world where whether we talk with someone or not, but we will put everything
This
on
is a
social media,
demonstration
like
of
Twitter
Wirecast.
or Facebook. So everything is there. So sometime the cops has not to do anything. They have to just watch the Facebook page of an individual and catch the criminals. So that's the state we are entering in. So that is basically happening is because we are getting isolated in the social setup. So that's itself is an issue, but it is a different issue. But you see, like why these social media people are
This is a
contributing to this
demonstration
thing.
of
So
Wirecast.
they have to make a balance between how they are making the money and how they are worried about the society. So the reason I want to make a bridge is because later on you will realize that we talk about genomics, we talk about the drugs, but we have some social responsibility. We have responsibility towards the patient. We have responsibility to the population. So that we need to fulfill. So that's the indirectly is the way. So the Bill Gates, like he does a lot of he donates a lot of money even in India, like
Wirecast.
vaccine efforts, tuberculosis effort. So he's donating a lot of money in those areas. Then this is another company, Consel, BRCA1, BRCA2, but I will discuss myriad genetics case later on, either tomorrow or day after tomorrow, that how they are BRCA1 and BRCA2, what is their patent controversy was. Now Gene by Gene, Laboratory Corporation of America, my map my genome is a Hyderabad-based company.
Of Wirecast.
Positive Biosciences is from Mumbai. So what they have is like they are doing the BRCA1 and BRCA2 third party testing. That's in India. That's very popular. Like you make a collaboration with a US company and you copy whatever they are doing is and then have your name coming. That's what they are doing is. So it's a Mumbai-based company. So basically they are mostly famous for BRCA1 and BRCA2 mutation identification. Then Quest Diagnostics, you must have heard this name, Quest Diagnostics. So they are also doing this. So just kind of there are several small players, but I just want to bring in these two companies from India. There may be small players which probably maybe just trying to exist or something. So that may not be here. But having said that, these three companies basically, they are the key players in the world. Okay. Now, when we are looking at the effect of the drug, so what we are looking at,
of Wirecast.
so let's say this is a line that is showing like how the drug is affecting the disease. Now this is benefit, this is harmful. Now if you go this thing, it is neither harmful nor beneficial. This is in other ways the dose. Now here it can be a mixed, it can be harmful, it may be beneficial, it may be 50-50, it may be 60-40. But here more beneficial and minimal harm. Okay, so this is the drug response that we are saying. So this is kind of a hypothetical curve. So we don't need this thing. We may need in some cases this thing, but this is what our ideal situation is. But you will see like here also I am saying that minimal harm. I am not saying no harm. That is in any of the drug, whatever company claims, companies never claim that any drug is 100% non-harmful. So every drug will have some side effects, maybe just a nausea, or maybe just a loss of appetite. It's very simple. But there will be some uncalled or unsaid, unidentified harmful effects that will be there. But you have to look at what is their positive effect, how the person get benefited. For example, if a person is suffering from tuberculosis, okay, you have a drug of choice, but it induces a lot of other side effects. So what you do is say you let the patient's body build in the immune system and counter for the tuberculosis, and rest of the side effects will fade away.
This is a demonstration
of
Wirecast.
That's the philosophy here, including in the cancer. So what happens is if you give a chemo treatment, so you will see like all hairs are gone, patient is weak, patients will become obese, loss of appetite, body pain. I don't know whether you have seen or not. If you look at the patient who has a metastatic cancer, it's horrible. Like their bones, they will have pains in the bone. They cannot survive. They cannot like sit. They cannot lie down. They cannot eat. They cannot drink. They cannot talk. They will just keep on crying with the pain. So when you give them the chemo, so when after second or third dose, when chemo start acting, it's really horrible. So that's why mostly doctors try to either give them some sleeping pill, sleeping injections, or anesthesia, or something like that, so that they can survive. So that is the that is the part which basically is the negative part. But you have a lot of other effects from the chemo, but still we
This
go
is a
with the chemo and
demonstration
we allow
of
it to go
Wirecast.
because we are looking for the beneficial. Now, have anyone heard about DLBCL? There was one person who said they are working on leukemia lymphoma. DLBCL. What is DLBCL? Diffuse large B-cell lymphoma. It's very common in India. Okay, not only in India, everywhere. So what happens is it has 4 stages again. Lymphoma cells are formed in the bone marrow. So if you don't catch them, the person's death is
This
guaranteed.
is a
But if
demonstration
you
of
catch them,
Wirecast.
there is a if it is CD 28, I don't remember the exactly. So there is a surface marker. So there is an antibody directed against that, and that is coming from Germany, Boehringer again. You take one injection, and I think you repeat it for 4 or 5 times, and after every 2 weeks. So that is the dose, depending upon the severity of the disease. And the person can be 100% recovered. Okay,
This
so
is
that's
a
the way
demonstration of
we are going.
Wirecast.
Otherwise, like blood cancer is very difficult to treat, but then the doctors monitor it first. Firstly, they call them after 1 month, then 6 months, then 1 year, then after 2-3 years they will say, okay, now you let it go, do it. And there are no restrictions, no diet restriction. If someone is smoking, they will simply say you lessen down the frequency of smoking. If someone is drinking, they will not recommend, but they will say, okay, if you are doing it, you cannot stop it, just keep it to a minimal level.
This is a demonstration of
So that's the way
Wirecast.
like our science is progressing, and there is another therapy that is coming up that is not in India, that is CAR. Against CAR is another antigen that is under clinical trial at this time. So if you use the antibody directed against CAR, that can be much more beneficial than whatever I have told you, CD28 antibody can be. Sir? Yeah. So this is a CAR protein that is sitting on the surface of
This
those
is a demonstration of
your
Wirecast.
lymphoma cells. That's where it is. So the challenge in the personalized treatment is, so we are talking about this personalized treatment. So the question arises who to treat and what to treat. Okay, how to treat. So means like firstly we have to find out a subset of the patients which needs the treatment to be further investigated. Let's say you give chemo and those patients are relapsing, or those patients cannot be
This is a
cured by that
demonstration
chemo.
of
Then you have
Wirecast.
to look at the alternative arrangement. So that is the challenge. Like how you, so that's where this biomarkers, as we discussed in DNA methylation biomarkers, you can combine the chemo versus neoadjuvant therapy can be used. Then second question, biggest question came is how to treat. Like, should we just give another chemo, or should we just give a surgery, or should we just treat it with another antibiotic, or what we should be doing is. Now, if you look at, this is the old scenario. So what happens
This
is,
is a
let's say if
demonstration
if
of
have a fever, for example,
Wirecast.
I have a fever. So this has happened with me, like not too long back. So I went to doctor. So they said, okay, you take this antibody, this antibiotic, okay. So he made combination antibody, but after 2 days, like I was kind of literally having a lot of body pain and everything going on. So we consulted another doctor here, okay, when I came back here. So he changed the antibiotic without touching me even, okay.
This
So
is a
I have to take it.
demonstration of
So
Wirecast.
I'm taking it. So this is the way it normally happens. So you go to the doctor, you say, okay, I have a fever. So they will ask you some general socio-economic question like, do you have mosquitoes around there, or do you have, what did you eat? Like stomach is upset, or like, is anyone infected in your home, kid or mother, father, or whatever it is. So just based upon that, he will decide, okay, and give you a broad spectrum antibiotic. Okay, so that's the end of it. Now then you go
This
back
is a
after a few days, you say,
demonstration
no,
of
no,
Wirecast.
doctor, I am not recovering it. So they will switch the amoxicillin to gentamicin, or gentamicin to erythromycin, right? It will keep on going. So finally what will happen is something will act and it will treat you. Or if it is a viral, it will automatically go back out of your body within 5 days. Okay? So it may be that you are just recovered from that cycle, or any one of these antibiotic has recovered you. So, but now with this new system, what we are
This is a
doing is
demonstration of Wirecast.
first of all, like we need to find out the reasons that why a person is predisposed to a particular infection or particular disease. Then diagnose it, like do the strep test or whatever other tests are, blood test or urine test or sequencing or whatever you want to do is, and then basically find out the right drug. Okay, so as I mentioned to you, there are
This is a
databases
demonstration
that
of
are built up, like
Wirecast.
CDT database or FK-based database. So you can match the history with those databases. It will tell you, okay, if this person has a history of with these symptoms and these sequence dysfunctions, so then basically this salt, it will tell you the salt, and then clinician can decide, okay, how much dose, how much this can be. So this is it is not an alternative of the clinicians. It is an aid
This
to guide
is a
the
demonstration
clinician to
of
decide
Wirecast.
dose and what drug has to be given. So this is basically the summary slide. So like basically we are in the cancer center. So the dream is, so I'm still giving you a term dream. So the dream is like whenever anyone comes, you have clinical symptoms. So the clinician will write down whatever swelling, blah 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 things are, and then basically you find out drug resistance, lifestyle,
This
nutrition,
is a
aging, genetic
demonstration of
variation. So you will
Wirecast.
find out the RNA sequencing, clinical features, genetic variation, then proteomic profile, as well as its any allergies are involved in that one. Okay, so and then you have to see that now everyone has to talk with each other. Okay, so one of the biggest problem arises is at the clinical stage. Okay, so when they are
This is a
diagnosing it,
demonstration of
everyone uses their
Wirecast.
different terminology. Okay, sometimes you must have seen that doctors has a very lousy way of noting the things like DT something. They will make up something, and then like if someone else is reading, you cannot make it out what the hell was going on. Okay, so now US has shifted to a digital electronic medical record. Okay, so what they do is that they just speak. Okay, yeah, this is what is. The person is suffering for 4 days. He has a
This is a demonstration of
asthma history. His
Wirecast.
mother was having this asthma history, so I suspect this might be the case. So that all recorded, and that is noted down in a digital database. Patient can see that, doctors can see that, or any if you change that PCP means another doctor, they can also see that. So they are moving into that, but still like it is taking a little bit of time in implementing on all the hospitals and with all the insurance carriers. So ultimately at the end of the day, it comes to the insurance companies, whether they want it or whether they don't want it. So that is a good
This
progress.
is a demonstration of
And being in the cancer
Wirecast.
center, that's what I was saying is, like our dream is, okay, anyone comes in, we will sequence it, okay, RNA-seq, DNA-seq, as well as proteomic profile and everything. But most of the time that never happens because everyone cannot afford that, okay. We have to charge the money from someone, and no company wants to finance that one. So it is still like a dream, like everyone will get done, but there are few good patients like who basically
is a
has
demonstration of
innovation back in
Wirecast.
their mind. They volunteer, and then there are a lot of clinical background data is integrated, and you can reach at a conclusion. But still, it has not become a custom practice that everyone will be getting sequenced. Now you have to understand this thing is I was planning to discuss it separately, but now if you sequence someone, let's say I have sequenced RNA of someone, So,
This
or like
is a
if I
demonstration
if I
of
sequence someone for
Wirecast.
the genetic mutations, and I found out that BRCA1 and BRCA2 mutation is there, so that means insurance company will say that no, we are not going to finance this person because she is already predisposed to preclinical conditions. Okay, that happens. Like you buy, so you must have seen like whenever your parents go from India to US. So there are some insurance companies, they say, oh yeah, we will provide you the insurance, you give this money and this money, blah, blah thing. So what they do is they put one clause.
This is a
One
demonstration
clause
of
is the patient should
Wirecast.
not be predisposed to the existing condition. Now if a person is more than 70, 75, 50%, 60% of the chances are they are predisposed to one or two, more than two conditions, and they never fund it. Okay, so that is the catch here. You know, they will say, yeah, give us 5,000 rupees, and we will fund it. Everyone is happy, parents are happy, other person is happy, but that is the clause. So that's the problem here. Then second thing is,
This
it may
is a
be that person
demonstration
is
of
getting
Wirecast.
isolated in the society. Like if you find out that person has leprosy predisposal genes, then probably that person will be isolated in the society. Then the third thing is, it is a social stigma. For example, you must have seen that if an HIV infected person gave HIV to several other people. So, you know, like, so those are the responsibilities of the patient to disclose those things, but they don't disclose.
This
Or
is a
insurance
demonstration
company
of
has their own
Wirecast.
stigma. Then society has its own stigma. So there should be something going on. US has done something which we call, I will discuss it a little bit later, which we know as a GINA, G-I-N-A. I think I gave you the reference. But before we go there, SNP we already talked about, single nucleotide variations. So basically in the SNP what will happen is, so one or two base pairs may be changed. Okay, for example here we are saying that
This is
these
a
bases are
demonstration
changed.
of Wirecast.
Now, when these bases are changed, if they are in frame, if they are in frame, but they are introducing the terminal codon, they can terminate the protein immature. So it can be, that can be lethal. So the protein is disformed, so that protein is not active at all in the system. So it can be like various ways. So these are two examples. SNP
This
in
is
beta
a
globulin,
demonstration
so it can
of
cause
Wirecast.
sickle cell anemia. And I will not go into details of this sickle cell anemia. You can Google, you can find out. So BRCA1, we will discuss it in details. Like if the BRCA1 and BRCA2 mutations are there in specific loci, so then those patients are predisposed to breast cancer. So this is what happened is, so this is the linked SNP, okay. This is the regular sequence. So the problem comes is your coding sequence is sitting here. Your SNP is coming here.
This
So
is a
what you should make it
demonstration
out?
of Wirecast.
Okay. Now whether this SNP is linked to this or this SNP is linked to this. That's why when we identify the SNPs, the functional studies are very important. Without those functional studies, we can just say like it may be, you see there is a logarithm linkage disequilibrium that we normally do in the SNP analysis. So it may be it gives you a good p-value over here, but not in here. Okay. So, and moreover, like it can, if it is near, it may not be
This
the
is a
causative agent, it may
demonstration
be
of
the causative.
Wirecast.
It can go in either way. Okay. So SNP can lie like several thousand base pairs apart, but still it can be affecting that particular gene. So unless we do the functional studies, there is no way like we can find out. A few examples like how SNP can. Now we are talking a lot about the mutations, variations, SNP. It's all in the one thing. Okay. So you must have heard one medication that is tamoxifen. Anyone knows about how tamoxifen? Where
This
tamoxifen
is a
used?
demonstration of Wirecast.
What's that? What's that? Yeah, yeah. So it's pretty good drug for breast cancer patients in specific cases. Okay. So, but it comes as a prodrug. It is not active. So now, if you give this prodrug, and if there is a mutation in the gene, in the enzyme that can activate it into the active form, So in that way, it will remain
This is a
inactive. It
demonstration
will
of
remain a prodrug, so it
Wirecast.
will not show any activity. But if it is active, then it will fully metabolized into active tamoxifen and will show its effect. Then I think another example is uh, the uh, the gene, that alcohol dehydrogenase gene. So I think I discussed a little bit about that. So there are like several
This
isoforms.
is a
So one of the
demonstration
isoform
of
ADH1 is
Wirecast.
very important. So if there is a mutation, every person has a different tolerance level of the alcohol, and then basically how sooner that alcohol dehydrogenase enzyme can act upon the alcohol, it will depend upon the individual's genetic makeup. Okay, so that's why, like, I think like, the easiest explanation is like some people like drink too much. Some people, some individual may
This
take
is a
maybe very
demonstration of
less.
Wirecast.
Even then, that is harmful to them. So it varies from individual body makeup to body makeup. Now, what is pharmacogenetics? Study of the genetic factors that influence the individual response to a drug. Now, there could be two things. One is pharmacokinetics, another is the pharmacodynamics. Can anyone explain what is the difference between pharmacokinetics and pharmacodynamics?
This is a demonstration of Wirecast.
Okay. So the kinetics means how the drug is basically being degraded or metabolized in the body. Yeah, so that is like basically
This
pharmacodynamic
is a
like
demonstration
how
of
the
Wirecast.
drug can target a particular organ or particular locus or particular thing, so it can get accumulated. So that's basically the key part is. Now we can use pharmacogenomics. So when we are developing the drug and we introduce genetic factors, genetic differences in the population, let's say Korean population, Asian population, other population, what is the difference? And then basically we call it as a pharmacogenomics. So this is the definition. Change in
This
drug
is a
metabolism.
demonstration of Wirecast.
Dynamics is change in the drug targets, enzymes, receptors, or transposomes. So this is basically an example. Like if there is genetic polymorphism, it will change the pharmacodynamic, like how the drug acts on its downstream targets. So what, it can be receptor, it can be ion channel, or it can be immune system, or it can be enzyme. So that's the way how this polymorphism means like mutations or
This is a
selects or
demonstration of Wirecast.
can affect the pharmacodynamics. So basically, it's affecting the response of the drug towards the disease treatment. Targeted therapy is a different concept actually. So what happens is, say you are targeting, for example, you must have heard about the Herceptin antibody in the breast cancer. So what it does
This is a
it
demonstration
targets the
of
HER2
Wirecast.
antigen on the breast cancer. Now HER2 antigen is present only in the metastatic cells. So when the breast cancer gets metastatic, it converts into metastatic. So that is aggressive breast cancer. It will produce the HER2. Now if you have, let's say, tamoxifen, let's not go too far. You have tamoxifen, tag it with the antibody against the Herceptin, so it will specifically target those things. So we are what basically doing is, so I will not confuse it with
This
the pharmacokinetics
is a
and
demonstration of
pharmacodynamics,
Wirecast.
but that is a different approach where you are targeting a specific target. So basically you are doing a targeted molecule that your drug is taken there. We have basically, there is a report where what investigators have done is they have added the ricin, that poison, tagged with some cancer cell directed
This is a demonstration of Wirecast.
tagged with chemically tagged the ricin with that antibody and they are seeing that it's selectively bone marrow. It's a science paper. Very simple, no hi-fi science. Very simple. But that showed that it can give like a 60% of more survival than as compared to if you use ricin or any other drug as a without any targeted therapy. So those are the kind of different So this is example
This
of
is a
HER2
demonstration of
antibody, as we were
Wirecast.
talking about the targeted therapy. So it is overexpressed in 25 to 30% of the patients, and basically that can lead to the increased production of the cancer cells, and we can treat it with the HER2 antibody, and this can inhibit target specifically the HER2 antibody. So the advantage is you need less drug, and it is targeted to only selected cancers, not everything. So this has become very important.
This
like
is a demonstration
prevention
of
point view.
Wirecast.
So if someone has a BRCA1 and BRCA2, breast and ovarian cancer prone, so then generally like they can undergo some pre-diagnosed prevention measures from the story after once she got breast cancer surgery. So it basically bumped up all the way the personalized genomics into a different area because she said that her mother was having, her grandmother was having, and they died because of the breast
This
cancer.
is a demonstration of Wirecast.
Now, if you look at from the effectiveness, like the oncology point of view, we can do HER2, or we have the colon tumor, then rare diseases, cystic fibrosis, CFTR genes. So these are specific mutations that are present in the CFTR gene, or this is the warfarin dosing, like if this mutation is there, probably warfarin may not be active in those patients. So just kind of give you a, I think I will
This
not
is
go
a
into
demonstration
this
of
part.
Wirecast.
So at this stage where we are, so I skip the single cell genomics. So at this stage, what we are is like we have the public databases. We can have a TCGA,
This is a demonstration
ICGC,
of Wirecast.
DBGAP, and all that control, tightly controlled, the clinical coverage data is there. We can find the mutations, we can find out the non-synonymous mutations. Everyone know about non-synonymous mutations? Non-synonymous mutations. Synonymous and non-synonymous mutations. Assignment है ये, test में आएगी.
This
What is the
is a
difference between
demonstration
synonymous
of
and non-synonymous
Wirecast.
mutation? You will get this question in the test. Okay, be ready. What is the synonymous? Uh-huh. So basically synonymous is silent mutation, non-synonymous is
This is a demonstration of Wirecast.
yeah, pretty close, but I think like you need to go back and look into that, and I will try to put some question directly or indirectly from you. Okay, so when you associate, we have a lot of data from the GWAS studies, so we can correlate with them and associate with these, find out the non-synonymous mutation which are in the coding region and which can affect the protein. Then either we can have immunotarget them. So what happens is, so here's the important point.
This is a
What happens is
demonstration of Wirecast.
cancer can be treated, but your body, or let's say you must have seen in some cancers, the bone marrow again produces the cancer cells. So the reason for that is there are tumor initiating stem cells within the bone marrow. So that has a signal from the cancer cell. So that will again proliferate back into the cancer cell. That's why the patient will relapse with the new cancer. Even if you have a chemo surgery or anything like that.
This is a
And then
demonstration
basically
of
you can
Wirecast.
screen for those, like to stop this tumor initiating stem cell, we can use FDA's um, catalog of compounds. So what happens is anyone find a new compound, FDA keeps on cataloging those compounds with their chemical features. So you can find out which compound may be useful by in vitro cytotoxic studies, lab studies, or other things. And then it can go back to the patients.
This is a demonstration of Wirecast.
So this is what I was trying to say is like, now I think this is a little bit important too. Like, if someone gets sequenced, so we are dreaming of a world where you go to a pharmacist and give your sequence, and he will be able to match it and tell you this is the drug that you are going to give. But will that is ethically correct or not?
This
So
is a
what will happen is
demonstration
you need
of
to
Wirecast.
have a consent from patient, from the other bodies, like they should know it. It might be possible, like once the social media has come into that much of the popularity, it is responsible for several divorces in the developed countries. Okay, so you can very well imagine like how much information you need to put it on the social media. That is also like in your hand, but you are putting it.
This
But that's
is
the
a
way the things
demonstration
are.
of Wirecast.
So how much openly information is available? Do you want the sequence should be available to everyone? Probably not. Then, so this is very important, which I was talking about, the CLIA lab. There should be a formally accredited lab. Now it may be possible if the lab does some mistake in identifying some mutations or some other thing, so it will be life tagged with that particular patient. So those cross contamination and proper accreditation
This
of the
is a
lab
demonstration of
must.
Wirecast.
Then interpretation should be done by a qualified person, maybe clinician or non-clinician or whosoever it may be. Then there should be consumer protection legislation to prevent false or misleading claims. So this happens a lot, at least in the United States. Like you say that, okay, you gave me false information, and that's why, like for example, if a person, I will give you a very
This
crude
is a
example.
demonstration of Wirecast.
Let's say a husband is infected with HIV, so he's supposed to, if he marries someone, he's supposed to tell before marriage that, okay, I am HIV infected. Okay, if he does not tell, then the woman can sue the husband at a later stage. And same is true for women also. Like she has to explain and all those things. So there are a lot of litigations that going around in the world. Like there should be transparency and there should be ethic regulations like what you do. So now the question
This is a
arises is that
demonstration
we were
of
discussing is,
Wirecast.
like we are talking about a lot about, okay, we do the sequencing, I keep the sequencing, I have access to everything, and I can do anything whatever I want, but I can sell it to the company. So but mostly this Google, this Twitter, Facebook are doing is, so in other words, Google, this Facebook, Twitter, these social media were created by the government at one point in time to like
This
track
is a demonstration
different
of
agencies.
Wirecast.
Okay, so but once they make it open, so now like everything is open, so they need not to go anywhere. Like if you see like any good agency, they don't do anything. They just track. Okay, this is the case. This is the Facebook account or Twitter account of a person. So they will make the file every day, and they have a psychologist working them, so they can make your mental chart, like what
This
what
is a
kind of
demonstration
tendencies you
of
have,
Wirecast.
and then they can follow you. Okay, so it's good, it's bad, either way, whatever the way you want to think is. So that's the way these things are. We are now, but it can happen is like if someone's sequence is exposed, as I mentioned, if someone is BRCA1 mutation is there, so that person can be neglected. Insurance will say, no, I'm not going to finance you because it's going to cost us millions of dollars or something like that. So then George Bush basically signed
This is
after
a
10
demonstration
years
of Wirecast.
tough negotiations with different parties, this act, which is known as Genetic Information Non-Discrimination Act, so which is basically says that insurance companies, based upon the predisposition of a disease, cannot neglect to insure a patient. Then secondly, like the patient cannot be outside
This is a
sidelined just based
demonstration
upon
of
the
Wirecast.
predisposition of the disease. Then it also prohibits, like the people, for example, like I have access to all these databases, so I can figure out, okay, this person is, I showed you data like a person died of IHED sometime back, a day before yesterday, but I can leak that information, so it comes under HIPAA regulation, what we call it, okay, that you cannot just release the information freely to all the world.
This
and
is
their
a
family
demonstration
gets it.
of
So
Wirecast.
there were like strict regulations. Then again, it also limits the employer's right to request requirement. For example, it may be possible. So what happens is, say, if someone die or gets sick in the job, then his or her employer is supposed to cover to certain extent their expenses. Okay, so they can say no because you are already predisposed to, so they cannot refuse that. So these are the nice regulations that are in
demonstration
place
of
before
Wirecast.
we actually get going with the personalized genomic medicine. So now what next? Okay, so we have been talking about genomic drug, pharma industry is basically interested in making more money. So what basically we do? Okay, so now genomic drugs comes in, FDA has to regulate, it has to go to the pharma industry. Okay, what we need is, we lack a bridge between genomic
This
drugs
is a
development
demonstration
at the
of
clinical level,
Wirecast.
academic level, and the pharma industry, and then FDA. So basically, there is what we call it the regulatory bioinfo, and I think, like, to a certain extent, we are working on that, making a group of people who will be coordinating with FDA on regulations, that what regulations can be done. So when you are working with clinicians, you have to remember that most of the time, clinician has their own way of doing the things. They won't listen, okay? Because
This
they
is a
don't have the time.
demonstration of
They just
Wirecast.
keep going, okay? So, but the pharma industry, they will always look for the profit, money, from where they can make. They care about science, but they will care about, okay, like if this thing I get it, I invest $1 million, how can I make $20 million from that? Okay, so, and if they are not sufficient patient, probably they will not have interest. If there is selected population in one state, they may not have the interest. Okay, so like you have to
This
bring
is a
them in and
demonstration
then
of
fight with FDA,
Wirecast.
and then so this is the body. Probably we are assuming that would make a bridge between these agencies. Okay, so we will continue with the regulations on the patent issue. Basically, R is nothing. R is just a statistical language, and which is kind of freely available. So the key
This
part
is a
in R
demonstration
is,
of Wirecast.
it is a community effort. It is not controlled by any company. It is not controlled by any academician, or it is not controlled by any specific group or anyone. So you must have seen like SAS, SPS, then MATLAB, and then there are several packages. Most of the bioinformatics is done downstream is by R. Like once you have alignment and other things, or like for example, like I have to download
This
a data
is a
set from the
demonstration of
TCGA, then
Wirecast.
I can write the script either in R or in Python. Okay, so basically R replaces most of the languages. It is not as efficient as like Python, C++, Java is, but it will keep you going whatever you want to do. So again, it boils down to the same point is like it is not the end of the world that if you don't know Python, it's not the end of the world, like if you don't know C++.
This
There are
is a
several ways.
demonstration of
Whatever works for you,
Wirecast.
just do it. The thing is that it should be done. That's it. So basically what we do is say we can have a bridging script which we say that okay, you go to the now how to download the database data sets from the public database. So what happen is we normally do it through the HPCC nodes, high performance computing nodes. Now when the nodes which we run,
This is a demonstration
are-
of Wirecast.
they are not on the net. They are not accessible to the outside environment because of the HIPAA regulations. But there are head nodes. For example, like when I log into like punch password, when I go hit there, so that is the head node. Now that head node recognizes the net, so it is exposed to the outside world. So whenever I write a script, I will run it over there, so that it can talk to negotiate with the
This is a
online,
demonstration
with let's say
of
TCGA
Wirecast.
dataset, ICGE dataset, or any databases, and then download it in a directory wherever I specify that one. So so basically, what is R and setting up R and R Studio, few calculations, handling of the files, some basic statistics, and how you download and use the packaging and use it for different writing functions. So R is nothing but it
This
is
is
a
a
freely available
demonstration of
language.
Wirecast.
So it is much like Excel and SPSS, SAS, but it is different. Okay, so I will not buy this idea that Excel and R is same. Excel does lot of mistakes. For example, it will take the month name as a like if you put like like 10/8/2017, it will take it as a month name.
This
It will,
is a
it will make it as
demonstration of Wirecast.
August 10th, 2017. So that's the way it changes the gene names. Okay. Then the macros which we write in Excel, so sometimes they are useful if the data set is small, but sometimes they do mistakes. But Excel is very well known to do some of the mistakes, and Microsoft is aware of those problems, but they have never fixed those problems. So that's why, from the bioinformatic point of view,
This is a demonstration of Wirecast.
yeah, but still there are problems. Yeah, still there are problems. I know, but you can you can format the column, you can format the row, and you can make it like numbers, and then it will pick up 0.000 numbers. Okay, so from the bioinformatic point of view, we never recommend using Excel. That's the bottom line. Okay, we don't
This
use
is a
Excel.
demonstration of
But
Wirecast.
you will see when I talk with biologists, I normally use Excel because that is easy for them to understand it. So at the most what we use the text files. Okay, so text files keeps the contents the same. It will not artificial changes, and then there are a lot of mistakes done is when you have a macro, you push the macro from down. So sometime if it misses one column, it basically disturbs all the
This is a
calculations.
demonstration of
So-
Wirecast.
It's a very famous saying in our USCs, and it's outside our door, that don't use Excel. So if you want to work, then firstly stop using the Excel. Okay, but I'm gonna say again, that's the one thing is, but for like biological labs, you normally do Excel, and when the companies send you the data, they always sell in Excel or in the text file, green and red, that is marked. So it's easy to discuss with you. For that matter, we use it.
This
But
is a
technically speaking,
demonstration of
I would discourage you
Wirecast.
to not to depend too much on the Excel. SAS, SPSS, these are the statistical softwares, which behind the scene, they have the packages same built in from the R. So what mostly MATLAB is a very classical example. So what they do is they pick up that package, make its help file, and use it for the commercial purposes. Okay, so if someone does not want to do lot of coding and other stuff,
This
they can
is a
use
demonstration
MATLAB.
of
Very
Wirecast.
nice, elegant, and easily. But if you want to introduce your some part in the coding portion with the already existing package, you can also do that. You can make a bridge. So that makes your life more easy than if you are working with the R console or R studio. The problem, the caveat here is, so basically in R you can play with your data,
This
you
is a
can find out what is
demonstration of
going on,
Wirecast.
you can read it, you can write it, and you can look at if there is a biasness, you can do some basic functions in R. Now, why we should be using R? Why not be using the SAS, SPSS, or Excel? So they have a limited ability to a different environment. So this is a nice way of saying that they can encounter the environment in a limited way.
This
But in
is a
R, there is no
demonstration
limit.
of Wirecast.
Okay? So then mostly in SPSS, SAS, MATLAB, you pay the price. Either your institution pay the price or you individually pay the price. And then you have to understand that, for example, like I also use MATLAB, I also use SAS, I also use R. I also use Perl, I also use Python. Okay? There is no binding that I am allergic to one.
This is a demonstration of
I am
Wirecast.
favoring to the other one. At the end of the day, so what you are looking at is how your things get done. Okay, so what will happen is, if let's say MATLAB has introduced a very simple hierarchical clustering package into their GUI interface. Okay, so in that what you will have is you will have a CSV or text file, and then import it just like you are doing in the RStudio or R, and then there are options like what you can choose.
This is a demonstration
What you
of
can do is
Wirecast.
but then in the end, like you become limited. Whatever it has given, you have to be satisfied with that, but not with R. Okay, and that's the whole purpose of doing this exercise. Like you can color. I have like a three clusters, if I'm not mistaken, or 4 clusters. So I can color the clusters differently. Put the same bar over here, which you will spend like several hours if you try to do it in Excel or PowerPoint. So it will just automatically output it,
demonstration of
and then you will get
Wirecast.
very high quality figures like PNG or PDF file, or you can also have SVG file. Are you aware of SVG files? SVG files you can play it in Adobe Illustrator. So you must have seen people publishing some of the files. Like I think I might have shown you those green, red, and all those different colors, even in the IGV or like others. So those are all the files that have
This
been
is a
edited in the
demonstration of
Adobe
Wirecast.
Illustrator. So there is a peak. You have to learn the Adobe Illustrator a little bit. You have to play with that, and then you can do a lot of wonders with Adobe Illustrator and then bring it back. Even you don't need Photoshop if you are working with the Illustrator. But if not, Adobe Photoshop will give you whatever you want to achieve. Okay, so so there are several functions
This is a demonstration of
in R,
Wirecast.
which, like, I think don't get confused this R with statistical language. R, that's the nomenclature that is given to R as a R statistical language, but it is in true sense it is not a mathematical or statistical language. It is basically, it is meant for doing any job whatever you think. Yes, it will do all those Wilcoxian rank test and all those statistical packages are
This
there.
is a
But if
demonstration
you
of
want to do
Wirecast.
good graphics like ggplot or hclust and all that, so you can do that. You can have a nice graphics. So one of the best graphics that you see in the publication, they come from R. Okay, so that's very important. So don't get confused. But this package, this R, was originally discovered by 2 friends who are statisticians. That's why its name is R statistical language. But in actual, it is beyond statistical language.
This
Statistical
is a
language is
demonstration of
one of the components,
Wirecast.
but it can do all the wonders, whatever you think about. Now, since this is a community effort, so what happened is that they have made different centers. For example, we in nearby in USC, we don't have this R special center, but UC Riverside, University of California Riverside is there. There is one guy, Thomas Geik. So he is very famous in R. You can
This is a
Google him or
demonstration
find out.
of Wirecast.
So he does a lot of stuff for R. Okay, so he runs R workshops. He writes several packages. He has several people working for him. So basically, he standardizes a package, take it to the CRAN package, submit it. Not only he, like everyone. Like if you write a package and before publication, there is a developer area, you can put that one, people will use it, give you feedback, you improve it, and once it qualify the CRAN criteria,
This
like
is
now
a
there's no
demonstration
bug
of
is there, so it can be
Wirecast.
used, just put it on the open, it's open. Developer is also open source, and then once the complete package is under CRAN, that is also open source. Source is available. That what you in the MATLAB or others you are seeing is, you are seeing like nice graphics, picture is coming in front of you. You don't see the background. Background is this, okay? In most of the cases, this is R that is running. SAS is also, SPSS is, there are very few packages which are their own. But mostly
This is a
they either
demonstration of
like
Wirecast.
derive it or change it. Now, here another important thing is probably which I would like to share is, you must have seen some of the commercial companies, for example, Avidis, someone was telling me, or CLC Genomics, okay? So they will bring those their own analysis pipelines. But what they will say is, so here is the catch. So they will say, we have developed our own aligner, okay? They have not developed anything.
This is a demonstration
Trust me,
of
they have not developed
Wirecast.
anything. Behind the scene, they are using BWA, Bowtie, or Top Hat, HiSat, or all those. They just change one line code. That's it. Nothing else. So they are saying maybe mismatch penalties by default, they have changed other things. It's nothing going on because I'm saying if commercial companies start creating their own, Novelgen created their own aligner. Novelgen is a company, but they could not sell it too much, like only 10 or 15. That's it.
This
So
is a
then
demonstration
people will
of
keep on coming,
Wirecast.
asking you the question. So in those scenarios, these open source tools or open source codes are pretty useful. You change the code or you do whatever you want to do is, and then come back and put those questions and answer on the forum and tell the people this is what you have done, this is what you want to do is. So now the same is like any other company. I don't want to criticize one company, single out one company. Partech is also, they also
This
claim
is
like,
a
oh, we have
demonstration
done
of
it. They just change a
Wirecast.
little bit. Basic concept, Smith-Waterman alignment remains the same. No change. Speed might change, 5 minutes here and there, maybe couple of thousand more alignments, and that's it. Nothing else. But at the end of the day, you will have the same alignment. However, I would also like to caution here, you will need a lot of publications where different computer programmers has come up with, oh, we
This
have
is a
developed a new
demonstration
aligner,
of
title is
Wirecast.
this, but if you read it, let's say they have made some changes to the BWA. So they will start comparing with BWA and blah, 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 blah things. So in the conclusion, you will find that, oh, they have said that in comparison to BWA, it used less computational power. Okay, that's it. Student has done the degree, but how much you care about that thing. So you care about sensitivity, specificity, and all those things. It may have 5% plus or minus
This
more,
is a
but if you are a
demonstration
good
of
bioinformatician, you
Wirecast.
can figure it out, like what is going on, even in like the original BWA. So that's why I have made it a philosophy. I may be wrong, that I generally stick with what are the basis of that aligner. I go by that. And then I make the changes if I feel there need to be changes. Instead of blindly following someone like, okay, Star has made these changes, or this has made some changes. So when the development occurs, So there are always new developments
This
that
is a
comes in.
demonstration of Wirecast.
But having said that, how much you are achieving in that and how much you are missing, you have to make a balance. So instead of going into those details, I always stick with the original aligners. And if I have a doubt, now for example, there can be a challenge, like there are 5 different aligners are there. So a lab will come and say that, okay, which aligner we should use? Okay. I will say, oh, I use it mostly. They will say, oh, no, we want to use this. What is the rationale?
This is a demonstration
Because our
of
collaborator has used
Wirecast.
that. Okay. So in those scenarios, you can use a simulation package. I think I explained it before. So you can have like all those 5 aligners running parallel, and then take a toy data set and see which one performs better. Now performance, again, it is a hypothetical question. What you are looking at, are you looking at sensitivity? Are you looking at specificity? Are you looking at unique alignment? Are you looking at like how much gap penalty you're giving? Are you looking at
This
how
is a
much
demonstration of
minimum
Wirecast.
mismatch base pair you are giving it? So it depends upon, again, the question you are asking. I'm not trying to confuse you, but I'm just trying to tell you is like what question you want to answer, you have to do that. But again, let's say you do the simulation, you have high set is or top hat is your aligner of choice, then you stick with that throughout your study. It's not that next time you come back, no, this data is good, you should do again simulation. Then remember that you have to compare apples with apples. You cannot compare
This
oranges
is a
with bananas.
demonstration of Wirecast.
So you have to have a consistency. If you have used Topet, then no matter your data is good or bad, you have to use that. Otherwise, you do everything from that. So if you start doing that part, probably you will not be able to finish at least one project. So this is the frequency which we normally, as from the bioinformatic point of view, will face from the biologists. Okay, so but most of the biologists, they understand like what is going on. But having said that, so all these little
This
bit of
is a
changes,
demonstration
whether
of
it's in the
Wirecast.
background, it is Python, Java, C++, Perl, or anything is, you can achieve all this with the power of R. There is a drawback also in R. Now this is the strength which first we want to talk is, so it can easily tell us data management and manipulation, statistics, graphics. It's a programming language, and it's free, and it's a community based. So you can have a nice graphics.
This is a
So as you see, you
demonstration
must
of
have seen several
Wirecast.
publications. So can anyone tell which package is used here? Yes, ggplot, what function? Can you? Scatter plot. Yeah, it's a simple scatter. Yeah. So this is the
This
same thing
is a
you do in Excel,
demonstration of
right? You have
Wirecast.
2 things, okay? You have 2 things, and you have 2 types of data. So you plot a scatter plot, and then you plot a line of regression, right? So it's the same thing, but see, nicely it has colored is, like this is the way this color varies, like everything is tightly knit in this one as you go spread out, so that the correlation or their similarity keeps on separating out. So that is nothing like basically
This
you are
is a
comparing
demonstration of
2 data sets
Wirecast.
under different conditions. Now this is again very nice graphic. So anyone want to tell what this? What's that? What's that? Yeah, clustering is there, but what kind of clustering is this? What kind of clustering is this? What's that? This is not a
This
PCA
is a
plot, at least.
demonstration of
It's not a PCA
Wirecast.
plot, but it is a one-way hierarchical clustering supervised. So why we call it a supervised clustering? Because here, why he picked up supervised clustering word? So because here we are annotating, I think it is not very clear, we are annotating whether this is tumor, which tumor sample is, which type of normal cells are, or which kind of like high grade, low grade, or and all those things are there,
This
and that's
is a
what they are marking.
demonstration of
So
Wirecast.
basically, what supervised is like we know the covariants, and we assign those covariants to different samples, and then try to figure out what is the trend. That is what we call it supervised clustering. So in our case, what you will be doing is that is the unsupervised methylation clustering. Okay, not the RPMM is the unsupervised way of clustering that we are going to do is.
This
Now
is a
weakness
demonstration
is not
of
very user friendly.
Wirecast.
There is a steep curve, so there is no commercial support, and it is slower than as compared to any other typical programming language. However, keeping in view all these drawbacks, R is still very powerful because of the open source coding and community support. It's a very good source of programming in the bioinformatics field.
This is a
So
demonstration of
if you try
Wirecast.
to in Linux, there are different packages that you install. So if you install this, the R, you will see like there are thousands of the sub packages that are installed in that one, and like so that basically depends upon how much usage you have it as compared to any other. What do you call them in the Linux? So this is how the R console will look like. So basically this thing, okay, this is what it is, the R console is. Okay, then you can have this kind of a partition where you see this is
a
the function that is
demonstration of
running this.
Wirecast.
Again, like I just don't want to burden you like what this is going on. We will discuss the specific and basic functions in the subsequent slides. But you can have this kind of thing. So if you can realize how much you can do this with hyphens, brackets, and all these things, you are good to go in R. They can do all the wonders for you. Where to put, close the bracket, where to start the bracket. So if you do one mistake, so this is another caveat
This
in the
is
R
a
is
demonstration
it
of
will give you
Wirecast.
generic error message. That is not very helpful for a non-R person. For example, like if I have assigned 3 clusters and I keep on going by mistake, like I am sleeping, so I click something, maybe bracket, I closed it after second cluster, it will take it, it will discard the other ones, and it will keep on going, and in the end it will throw the error that you don't have an exact functional whatever the structure of
This
this
is a
thing, it should be
demonstration
more
of
than 0 and
Wirecast.
everything. So it's very hard for a non-R person to find out. But yes, if the error comes in and you know specifically where the error is coming, you take that error, Google it, you should be able to find out in some blogs the questions and answers. So there is a R forum also, pretty good. But I would again encourage you to go for like R Biostar forum or sequencer forum.
This
So
is
they
a demonstration of Wirecast.
they are pretty good source of most of the stuff. But there is an R form also. Probably for a person who is not familiar with R, that may be too overwhelming. So another like drawback with R is if a person knows R, they talk in a different language, okay? Because they think that with my due apology, they think like they are superior than the others. Okay, this is a common. So they
This is a
basically talk and say,
demonstration
oh
of
yeah, you can use this
Wirecast.
function and this. So the other person will get intimidated. So when you ask a person the question, very specific, say that okay, write the function here. That's it. Okay, so that's the way you get your things done, because this is not something personal. Okay, it's just a habit they all develop. Okay, but you get your things done. So you say, okay, that's fine. I'm not very familiar. Can you just write the function here? That's it. Very simple. Okay, they will
This
write
is
it.
a
They will speak
demonstration
a
of
lot.
Wirecast.
Let it go. Okay, that's the way you get help. There is a lot of help in R, but I doubt that is very useful for a person who is not familiar with R. Okay, so that is if you know R, as you develop skills in R, there is a curve which takes a pretty long time. It's not that uh, that you just try it for a week or 10 days, you will get them going. And I don't think you need to worry about that. So the way I have done is,
Wirecast.
I have all the packages and I have all the scripts along with the toy data to be saved in different archives. Okay, so if I, for example, like if I have to do RPMM clustering, yeah, I can download the RPMM package, but there are 20 other things going on. Seriation package, this package, and then I have withdrawn it. Then I try to mark it as they can tell, like within the script, I have marked in the hyphen, like what this package is doing, like what is the normal mistake that can happen is, I think I must have mentioned, make sure that it is not like this. Some of the things you can write, and no one can remember that. There's no harm in that. And I shared that script in the same way with them. So, but that is the way you kind of make your life easy. If you just keep on hiding the things, oh no, what other person will think, like why I'm not doing this, then you will be the sufferer. You will waste your time. I will waste like my several days in finding out one package, reinstalling it, then error comes in, one package was made for 2.1.3, this is 3.1.0, and like I just waste a lot of time, and it is a job of 5 minutes. I will go back, but yes, you have your brain working, but I know why it's sitting in my brain that I know, okay, this data set, we use this, and it was somewhere in 2014. I will go back and find out where I have space. I will figure out, take out, redone it, and spend like an hour or so if this is giving an error, and then correct it, move ahead. So that's the way you will do with R. So there's no one who can memorize all these commands, but basic function is a different thing, like equal to, multiplication, scatter plot, like hierarchical clustering, and all these. These are the kind of basic things like everyone is supposed to know. But there, other than that, there is no one like who can remember all the commands by heart, like whether it is R, Linux, or anything like that. You can Google it, you can find out.
This is a demonstration of Wirecast.
So the latest version is 3.4.0. So you can go to the R project and just follow the installation instructions. Okay, you can go it over here and just follow the instruction. It will automatically pick up your computer, like whatever your Windows is, 64-bit or whatever is. Don't go for the binary part. That is for the developers, like people who know how to organize your package. So this is basically the R
of Wirecast
project. So if you see, it says this is a recent development that has been recently done, I think, in the month of June 2017. So you can download it. Now, another thing which I want to tell you is, sometime what happens is, say, it takes you to different mirror sites. It has several mirror sites. So the site nearby us is UC Riverside or Michigan. UC Riverside is again Thomas Guy's site,
Wirecast.
and University of Michigan is someone else. You can pick up any site, it doesn't matter, but don't pick up a non-English site where you have a language problem. Okay, so just pick up any site. I don't know whether, is there someone from India too? Have you seen any mirror site from India? Mirror site from India? Okay, anyway, so you can pick up any of the site.
This
and
is a demonstration
maybe
of
MIT or
Wirecast.
any good institution, because they will maintain it pretty well. And if there is an error, then their packages are pretty neat and clean. So this is again this is the sign where download R.
This is a
So you
demonstration
can
of
have
Wirecast.
Now similarly, you can download the R Studio. So there are two versions. One is for desktop, another one is for server, and you will be downloading it for the desktop. Okay, so it can have a
This is a
Windows, it can have a
demonstration
DMG
of
file for
Wirecast.
Mac, whatever you want to, and it's also good for Linux. You can install it in the Linux. So we normally install it R in Linux and try to use, and that's where we try to manipulate the power. That's the only alternative if you have a big file, because it will take the virtual memory from your hard drive. That's the problem. So one of the caveat with R is it
This
needs
a
a lot of
demonstration
memory.
of Wirecast.
It's memory hungry, basically, because it uses your local resources. It does not uses your net resources, but it will use your local resources. So if your machine is not sufficiently powered, you might have problem with the bigger files and doing a lot of computational. But for small data sets, that should not be a problem. So this is the way like this console will look like. You will have, this is the main, like what we call it as
This
the
is
R
a
console.
demonstration of
So
Wirecast.
this is where your script will run. So this is R console, and this is basically the script editor where you will plot where you will like have a text editor. You can change the R script, or you can just open the R script, and that will be here. And then this will be your workspace. And now here you can have help, plot, packages, whatever the packages you want to download, or your figures can display
This
or
is a
save
demonstration of
under
Wirecast.
this area. So basically, when you run anything from here, you do is control, enter. The way you will do is you will highlight this. Let's say you want to run this command. You will highlight this, control, enter. So that will appear over here, and it will move further. If there is an error, the error will pop up here. Okay. So the first thing is you may like to do is say, I think
This
do
is a
this.
demonstration of Wirecast.
I will not worry about playing with the internet, because so once you download the R Studio, before R Studio become functional, it will ask you the R console. Okay.
This
So where
is
is
a
the R console? So you
demonstration
have to
of
download the
Wirecast.
R console first, then R Studio, and then from the C directory it will automatically pick up the R. So basically, now you understand why I was saying is R Studio is nothing. It is just like a nice graphic interface which is allowing you to work in the R console. Otherwise, R console is intimidating. Like if you are not familiar with these brackets and other things, you will say, okay, what the hell is going on? What I'm doing is, okay. So that's the only thing is.
This is a demonstration of Wirecast.
Okay, I think. So I showed you like open file, how you basically you can go to the directory, open in R script. We have already done that part. Now this is basically a typical R Studio. Here is your script. Here is your console. Here is your graphics or packages and other help and other things. This is your workspace. Okay, so
This is a
the
demonstration
important
of
part
Wirecast.
is you can save the workspace. Okay, if you save the workspace, now another drawback in R is I'm a fan of using R because our life is dependent upon R basically. But having said that, once you close R, everything is gone. Okay, then you have to not re-download. The packages will be there in your C drive, but you have to do a command like library,
This
RPMM,
is a demonstration
open those
of
packages.
Wirecast.
Okay, you have to do all over again. So what we do is say, the way I do is say, my USC computer is not under rules by USC. It has been made exception. Is they will not force any ITS what do you call it, policies by which either Microsoft will push me to automatically update, where it will restart the computer and other things, because there is a lot of stuff
This
going on.
is a
So what we do
demonstration
is
of
say,
Wirecast.
we normally say, okay, these are the 4 now files which I have to play with R, then I will just keep R Studio or R open and try to work on that unless we communicate with someone and then questions come back, questions goes back, and only then we will try to restart the computer or close that window. It's not like, probably it's not that much for me, but we just try to be lazy. Let's put it in this way. But that's a big
is a
problem. So it's
demonstration
not
of
that
Wirecast.
once you have installed all the packages, you are done for your life. No. Every time it will ask you to do that again. The same packages will be there in your C drive. Okay, that's why I think I am here saying is, so R works good. So basically, it will take you from the C drive. All these packages are in the C drive, but you have to, let's put it in a simple way, unpack them so that it becomes the same workspace whatever you have previously used. So the alternative is
This is a demonstration of Wirecast.
you can save this workspace for a particular analysis. Let's say you are doing PCA plot. Okay, so you can